Honorable laureates, excellencies, distinguished colleagues, ladies and gentlemen, welcome to the Nobel Lectures in Chemistry 2024. So this year's Nobel Prize in Chemistry recognizes groundbreaking achievements in two different areas: computational protein design and protein structure prediction. So it's all about protein structures. As you may know, the polypeptide chain of a protein folds itself up in space into a complex three-dimensional structure. Now, the remarkable thing is that this 3D structure is somehow encoded in the sequence of amino acids that make up the chain. This finding that a linear sequence of amino acids dictates the 3D structure, it dates back to the work of Christian Amundsen, who received the Nobel Prize in Chemistry in 1972, so over 50 years ago. It meant that it should, in principle, be possible to predict the 3D structure directly from the sequence. Likewise, it should be possible to predict which sequences would produce a certain 3D structure. So it has long been a dream of biochemists to computationally solve these two problems. And it is clear that one needs to use computers to do it. The latter challenge, to predict sequence from structure, that's what protein design is all about. The idea is to draw a new protein, typically one that has never been seen before in nature, and somehow compute a sequence that will give this structure. The sequence could then be made in the lab, and completely new proteins with desired functions could thus be created. So this challenge was solved by David Baker and co-workers in 2003. They designed an entirely new protein with 93 amino acids and computationally predicted a sequence that would give this structure. They then made the protein and verified experimentally that the prediction was correct. The reverse problem, to predict 3D structure from sequence, is an astronomically huge search problem. And this was pointed out by Cyrus Levinthal already in the late 1960s. So progress in this field was slow indeed for many years. But Demis Hassabis and John Jumper and their co-workers, they solved it in year 2020 by ingenious neural network engineering, which resulted in the now famous program AlphaFold 2, and it was published in 2021. This was undoubtedly the real breakthrough in protein structure prediction, and AlphaFold 2 gave unprecedented accuracy. So this network is trained using the data stored in sequence and structure databases to discover correlations and patterns among amino acid sequences. And this allows it to produce accurate distance maps that can then be converted into 3D structures. We'll probably hear more about it from the laureates. So the work of this year's laureates has had an enormous impact. David Baker has opened up completely new possibilities to design proteins that have never been seen before, with many exciting applications in pharmacy, medicine, nanoscience, etc. And likewise, the work of Demis Hassabis and John Jumper has caused a revolution in structural biochemistry, and we now have access to predicted structures of all 200 million known proteins. And this is a phenomenal resource for biochemical and biological research, also with many new applications. So our first speaker, David Baker, he was born 1962 in Seattle, Washington, United States. He got his PhD in 1989 from University of California at Berkeley in California. and he is presently a professor at University of Washington, Seattle, and an investigator of Howard Hughes Medical Institutes. So please join me now in welcoming Professor David Baker for his Nobel lecture.
All right. Well, first of all, thanks everyone for being here. It's really wonderful. It's a great honor, and it's wonderful to see so many friends and family and colleagues. So today I'm going to tell you about de novo protein design. This is an artist's depiction of the interior of a cell in our bodies, and each of these colorful blobs is a protein, and each protein is carrying out a unique job or function. And indeed, in all living things, proteins are carrying out, they're basically the miniature machines that are carrying out all the important functions. And they evolved over hundreds of millions of years or billions of years to be really perfect at solving the problems that arose during biological evolution. But today, we face new problems. We live longer, so neurodegenerative disease and other problems now are arising. We're heating up and polluting the planet. And so if there was selective pressure over a really long time, new proteins might evolve to solve these problems, but it would take a very, very long time and not be very pleasant. The question the goal of de novo protein design is to make new proteins that can solve modern day problems, as well as the proteins that came through evolution solve the problems that were relevant during evolution. So first I need to remind you of a little bit of biology, and you already heard this, but I'll just give you the basics again. The genes in our genomes, each gene encodes one protein. It does so by specifying the amino acid sequence of the protein, which then folds up to the protein with which has a particular structure and biological function. So in de novo protein design, the flow of information is just the opposite. We start with a desired function that doesn't exist. We build a protein which we predict will have this function, and then compute the amino acid sequence this protein should have. And then this is a brand new protein, so there's no gene in nature which already encodes it. Instead, we make a synthetic gene, a synthetic piece of DNA, and that encodes this protein. We put it into bacteria, and the bacteria basically become factories for making the protein. We can get the protein out and then test to see whether it actually does what we designed it to do. And so today, I'm going to be giving you a number of different examples of designed proteins that we've designed to have different functions. And in every case, we've gone through this computation and then made a synthetic gene and then made the protein, and I'll be showing you what those proteins do. Let's see. So there is a huge space of possible proteins. And so there are 20 different amino acids, and a typical protein is a sequence of about 100 or more amino acids. And so that means the number of possibilities is 20 times 20 times 20, 100 times, or 300 times, depending on how long the protein is. And this is an astronomical number. And it's kind of depicted by this gray space here. Now, what's been sampled during evolution is a tiny, tiny fraction of the possibilities. And the proteins in nature are kind of indicated by these red ovals, because evolution proceeds incrementally. So proteins tend to be very closely related. The proteins in us are closely related to the proteins in other mammals and so forth. So it kind of bunches up a lot. So traditionally, and this is really up until the present, if an engineer or a biologist wanted to solve a new problem and they thought a new protein could work, the way it was done, and is still being done, is to look all through nature to see if a protein can be found that has that activity. And that's called bioprospecting. The other thing that's often done is to take a naturally occurring protein that does something kind of similar to what you want to do and make small changes to it. And so you get a protein that's very close to a naturally occurring protein, and you've hopefully modified it to do a little bit closer to what you want it to do. But what I'm going to talk about today is different. I'm going to talk about building proteins completely from scratch that carry out new functions. And the advantage of this is you can build the protein to do exactly what you want to do. You don't have to worry about what the closest that, that nature got, or try and modify something that's already very complicated. So I'm going to begin with a very brief history. The first stage in de novo protein design was understanding how to build new protein structures from scratch. And this really culminated in 2003 with the work of Brian Coleman designing a brand new protein which had a new structure and a new function. And since that time, a growing and now quite large community of people, really wonderful scientists, a number of whom, quite a few of whom are here, have been working to both design new protein structures, but also to design new protein functions, and in particular design proteins that solve current day problems. And in very recent years, this field has sped up with the advent, and you'll hear a lot more about this in Demis's talk, of deep learning methods. So I'm not going to tell you much more history in my talk. I'm going to basically sort of fast forward to the present. I'm going to tell you about the deep learning methods that we have been developing for protein design, and then I'll give you examples of what protein design can now do. So we've been developing generative methods for creating new proteins, and these methods, or the one I'm going to tell you about, called RF diffusion, is really inspired by methods for generating images. And these methods work by taking a large set of data, for example, images off the internet, adding different amounts of noise to the images, and then training a network to remove the noise. And once this has been done, if it's been done well, one can start with completely random pixelated sets of pixels, successively denoise, and generate an image that looks like it's image off the internet, but in fact it's a completely new image. And in the same way, we took all the structures in the protein structure data bank, and I need to highlight that everything I'm telling you about from now on absolutely relied on the work of the tens of thousands of researchers who solved the protein structures in the PDB, because that's the data that we use to train. And also the people who organized the protein data bank, it was really an absolutely invaluable resource. So we took all the structures in the database, we added increasing amounts of noise, and then we trained a network to predict what the denoise structure at each stage was. So at the end, just as in the case of images, we could start with a random, completely random image and generate something that looked like a very plausible image. We could start with completely random configurations of amino acids, progressively remove the noise, and generate a new protein structure. And we found that these proteins folded up pretty well. We could make them very well. We could make them in the lab, but they didn't do anything. Now, in the case of images, as you all know, you can you can guide or you can condition the generation process with, for example, a text prompt. So you can say, generate an image of a cat sitting on the table, and that's what you get. And we can do exactly the same thing here, because it's not very interesting just to make new structures. What we want to do is make new functions. So we can condition this protein generation process on the function that we want to make. And one way to do that is shown here. This is the insulin receptor. We can carry out this generative diffusion process in the presence of a target, like the insulin receptor. And during training, the generative method has learned that that proteins bind to other proteins through very shape complementary interfaces, and you can see that as this protein gets built up, how the shape match of this new protein matches the target. And we've now used this to design proteins to bind to well over 200 different protein targets. It's pretty much a solved problem now. I'm not going to tell you about all 200 of them, but I'm going to give you some examples of how one can use these binding proteins to solve problems in a wide range of different areas. And I'm going to tell you about 3 types of applications today: in medicine, in technology, and in sustainability. And I'm going to begin by talking about applications in medicine. So snake poisoning is still a major medical problem, particularly in developing countries. And snake toxins interfere with really fundamental biological processes. Now, an antidote for snake venom needs to be a very stable protein or set of proteins. It has to be cheap to manufacture, because where they're needed is in countries where there isn't necessarily a cold chain. You need them out in the field. You need to administer them almost immediately. And we thought this would be a good match for protein design, because the proteins that we design almost always are very, very stable proteins, so you can produce them in large amounts quite cheaply. And so here is a designed protein which is binding to the toxin, which is here. And all this work is highly collaborative. And so our collaborators in this case tested to see whether this designed protein could protect animals against the snake venom. And so in this experiment here, the animals who got the snake venom alone died almost immediately, whereas ones which got the designed protein were completely protected. And so we're very excited about this as a general way forward for generating anti-venoms that can be more effective and much cheaper than in current approaches. Now, a major problem in current medicine is inflammation, which is associated with many autoimmune and other disorders. And central to inflammation is a protein called the TNF receptor, and it's actually the target of many drugs currently on the market. And so here you see a diffusion trajectory conditioned on the structure of this target, and you can see how it's building a protein which is really exquisitely shape complementary to the target. And this protein is very effective at blocking inflammation in animals. So these are animals that have been exposed to an inflammatory treatment or inflammatory signal from bacteria. There's a very large amount of inflammation. Current drugs like Enbrel that are used to treat inflammation have some effect. This designed protein, because it binds so tightly, is even more effective. And so we're very, very excited now about making drugs to treat a wide range of autoimmune diseases and inflammatory diseases. Now, cancer, of course, remains a pressing problem, and we've been designing proteins to activate the immune system to treat cancers. And so this protein here, this design protein, is bringing together two immune receptors, and this results in a very strong activation of the immune system. And again, with collaborators, Mike and Stephanie Dugan, they have been looking at pancreatic cancer, which is one of the most difficult forms of cancer to treat currently. And what they find is that this design protein is quite effective, more effective than other known treatments in reducing the size of tumors in this model. And again, we're very excited now about getting this protein out in the world for into clinical development. Another important application of design proteins is to make antivirals for pandemic preparedness. So this is the influenza virus surface protein, and here you see the diffusion process generating a binder here. But here we've added an additional conditioning criterion, which is we want this protein to be an antibody, which is a particular type of protein fold. And we wanted to do that because antibodies, the pharmaceutical industry knows how to develop antibodies, and so they like to work with them. And so here you see an antibody-like structure being generated that binds to the target. And here I'm showing you an experimentally determined structure of this design protein bound to the flu virus protein. And you can see that in one of these colors, let's see, the experimentally determined structure is in purple, the design structure is in gray, and you can see they're almost identical. And that those of you who know about antibodies know that they recognize their targets through CDR loops. And these loops, which are being built completely from scratch, in addition to the docking mode, are coming about almost exactly in the design model. So I think that antibodies are currently discovered using by immunizing animals or pulling them out of people. I think we're now seeing a transition over to using these kind of design methods to generate antibodies. So another sort of increasingly important medical problem are neurodegenerative diseases like Alzheimer's disease, which are associated with the formation of long amyloid fibrils. And there are several proteins, among them amyloid beta or A beta and tau, which form these fibrils that are observed in the brains of people with neurodegenerative disease. So here what we've been doing is designing proteins that will bind to the disordered portions of these proteins that would normally interact with themselves to form amyloid. And these trap these regions so they can't form this extended amyloid. And this experiment, again with collaborators, is looking at if you simply take these proteins, put them into solution, they aggregate as the amyloid is formed. And with these trap proteins, this amyloid formation is completely blocked. So this is measuring the extent of amyloid or aggregation that's going on. And so we're very excited about these as a possible way forward with neurodegenerative disease. So there are many, many other applications we're excited about now in medicine, but I'm going to switch over and now talk about technology. So one general problem in technology is in sensing. How do we sense arbitrary molecules? And how do we determine sequences of things like DNA and proteins? And in nature, there are channel-like proteins or pore-forming proteins that look something like this, and people have adapted those for problems like sensing and sequencing. But they aren't really optimal for that because they didn't evolve for it. And so with Anastasia Vorobyeva's group, we've been developing methods for designing new pores completely from scratch, and this has the advantage that we can completely customize all properties of them, of the pore and so forth, to be optimal for the task we want to carry out. And this just shows you some examples here. These are 3 designs with increasing pore sizes, and the larger the pore, the more ions that can flow through it, and so the larger the current that is measured going through these. Now, to turn these into sensors, and we're very interested in constructing molecular noses, what we do is to design proteins that bind small molecules that we want to detect, and we can put these proteins on top of the pore, like shown here. And then when we put this combined protein into a membrane and we measure the current across it, we see a sizable current. When the small molecule is added, basically it closes up the channel, and that's shown here, and then the current completely shuts off. And this can be measured because one can measure electrical signals very, very sensitively. So we're excited now about being able to make very general molecular or electronic noses that can tell you what's in a solution by integrating the information from many different pores like this. Now, what you'd really like to do is to couple this kind of sensing scheme with electronics directly. What I showed you before was those were inserting into a hydrophobic lipid layer. So we've been working on methods for inserting these sorts of pores into silicon nitride chips, so they could ultimately be for example, in your cell phone to tell you what's around in the environment. And so this requires a totally new class of pore, which is an example of which shows here. We want things, of course, proteins never evolved to interact with silicon nitride or to insert into it, so we have to design new types of protein structures. And here are some examples of EM structures of sort of this gallery of pores that we've made to try and to approach this problem. And again, with collaborators, we have, this is from Jens Gundlach's lab at the UW. So here we have a pore in a silicon nitride chip. There's a lot of current going through. The design protein pore can insert in, and we get a very stable current. And now we can start modifying these pores, as I showed you with in a moment ago, by incorporating binding domains and other sorts of domains on the interior to be able to read out sequences and other properties. So another general problem in technology is targeting very specific sequences of DNA. So, you know, our genomes have lots and lots of DNA in it, and there are many applications, for example, fixing genetic diseases where you want to target just a very, very specific region of the DNA. And so to do this, we've been designing proteins which bind to DNA and have very high sequence specificity. So the protein shown here makes very specific interactions with the DNA bases that make it very sequence dependent. And this is now some experimental data showing that if you take this protein and measure its binding to DNA, it's very sensitive to any changes of the DNA sequence. So when the sequence is the red color means that the binding is weaker. So these sorts of designs can read out DNA sequence very precisely. And we're excited about using these, for example, to create new ways of smaller, more compact ways of fixing genetic lesions and other activities. So now we have a very exciting, Neil King had this very exciting result last year with the first approved de novo design medicine. And this is sort of an interesting case where Neil started off making these self-assembling nanoparticles sort of just to see if we could do it. And then after he had succeeded in building them, he realized that he could make vaccines out of them by putting pieces of viral proteins on them. So during the pandemic, he put some of a part of the receptor binding domain of the COVID surface protein and on these, and found that this elicited really, really strong immune responses. And this is now a clinically approved medicine. It's in use in, it's been approved in several countries. Here is the designed protein in a vial, and we're very excited about, a year or two from now being able to have many, many examples like this. Now, these sorts of nanoparticles are very useful also for delivery, but there's a problem in that if you want to deliver a lot of material, like say a drug to a specific site in the body, the size of the container matters, and with a bigger container you can make more. So this is an interesting geometry problem. The icosahedron or dodecahedron is the largest completely regular polyhedron. So if you want to, you can make it bigger. So this is 12 pentagons coming together. You can make it bigger by inserting hexagons between the pentagons, as shown here. This is just as in a soccer ball. But here you have a problem because you can see the angle inside the pentagons is different than the angle inside the hexagons. So we've been trying to figure out how to solve this problem, and there are basically two solutions I'm going to show you here. These are both electron microscopy structures of designs. In this case, we've solved the problem by using just one type of protein that can adopt different conformations in the different sites in this nanoparticle, allowing the interspersal of pentagons and hexagons, as you can see here. In this case, we've used different protein chains that sit at the different non-geometrically equivalent positions. And the advantage of this is that both of these methods, and again, this is experimental data, cryo-EM structures, give much larger particles than one gets. This would get with pure symmetry. This, for example, is one of the most widely used current delivery vehicles based on a naturally occurring virus, AAV. Now, everything I've told you about so far is kind of static, but in technology, we'd like to be able to turn things on and turn things off. And so to do that, we've designed proteins that undergo conformational changes and have two different states. So on their own, they fold up to one state, but then when an effector is added, they convert into another state, as shown here. And if we build now larger assemblies out of these, like this triangular one here, in the presence of the effector, these will now bend, and so we'll get, and now we can effectively convert, you can see when this becomes a right angle, we effectively convert a triangle into a square. And this works really nicely. We chose to go for these simple geometric transitions because it's very easy to follow by electron microscopy. So here's a design just like this one that was designed to be a triangle. Then the effector is added, and you see you get a square. And this is electron microscopy data. You can clearly see that transition. And here's another case where you have a square that's going to a pentagon when the effector is added. And for the biochemists in the audience, this is clearly allosteric control because this effector is not acting at the interface between these chains. It's acting quite far away and working through by changing the conformation. Well, that wasn't very particularly useful. It was sort of like that showed that we could build switches. But where might you want actually a switch? And coming back to immunotherapy, we might want to activate the immune system to treat cancer, but then we might want to turn it off because the overactivated immune system can be a problem. So here I'm going to show you an off switch. Here's a designed protein that brings together two receptors activating the immune system, as I showed you earlier. But now we've built in a binding site for an effector which turns the whole system off. And here's a cartoon of how it works. So here's the design, bringing the two chains together. This effector comes in. It causes an allosteric change, which kicks off this other receptor subunit. And this is, again, a wonderful collaboration. This is actually going on the surface of a mammalian cell. You're seeing these two receptors are labeled with fluorescent dyes, and when we add the drug initially, the two receptors come together, and then when the effector's added, they come apart, as you saw here. And so we can see the receptors coming apart when the effector is added. And this has a profound effect on cell signaling. So when we add the drug, this protein, it brings these two together. We get an activation of signaling. And then when the effector is added, we can shut it off very, very rapidly. And this is useful not only for shutting off the drug when you no longer need it, but for probing exactly what the effect of having these receptors together for different lengths of time is, which has been kind of an outstanding question in immunology. And the final area I'll tell you about is sustainability. And some of the most remarkable proteins in nature are enzymes, and these carry out chemical reactions. And the way they do this is they have these very large, complex structures, and if you zoom in, they have active sites where there are a few key amino acids that are mediating the chemistry and carrying it out on a bound small molecule. And so we can now design catalysts completely from scratch. We do that in kind of the opposite way. We start by specifying the active site geometry, what the side chains on, where they should be, and the small molecule or transition state we want to act upon. And then we use the diffusion process to build up a protein around that active site. And so this is kind of neat because we can explore in nature, and biochemists have been, and enzymologists have been doing this for many, many years. You just have one thing in your enzyme, and you can sort of tell stories about how it works. But with this buildup process, we can change what the active site, what the side chains are in the active site, and what their geometries are, and look at the effect on catalysis. And so we can build up sites of increasing complexity, and what we find is as we adjust the geometry and make the sites more complicated, we get increased activity with a partial site. We get enzymes that carry out a single round of the reaction but then stop. If we build up full active sites with sort of more extensive networks of interactions, we get quite active enzymes. And we can solve the structures of these by X-ray crystallography, and then we can see to check to see how accurate our designs are, and they can be extraordinarily accurate. This is now a superposition of the experimentally determined crystal structure and the actual design. And the activities of these are much higher than had been found previously straight out of the computer. That was for a complex multi-step reaction. We can also use metal ions to carry out catalysis. Here, this is actually starting with a quantum chemistry calculation of the reaction pathway. And we identify the transition state on this pathway. And then we basically build a protein around the metal and the binding site. And this is sort of a blow-up of the site. And as I said, in all cases I've shown you, we are making synthetic genes, encoding the designs, putting them into bacteria and testing them. Here we tested 96 designs, and we got a number of active designs, the most active of which had very, very much higher activity than anything that had been made straight out of the computer before. And this is starting to approach the activities of native enzymes for this type of reaction, which have evolved for hundreds of millions of years. So there are many problems. This is a bond-breaking reaction, and there are many problems where this is important, including plastic degradation. So we're now building sites, completely synthetic enzymes, to break down plastic and other polymers using exactly this type of active site. Now, one of the other remarkable things that proteins do is to harvest solar energy, and this is done, of course, in the photosynthetic process, which is very complex, which involves very big proteins, but they have at their core what's called the reaction center, which is made up of 2 chlorophyll molecules. So we've been, again, kind of building from the outside, from the inside out. So here we've designed a small protein that binds 2 chlorophyll molecules, exactly as in the reaction center. And now we can start building this into larger assemblies. And so this shows taking this protein here and now designing it into these cubic assemblies. Each one of these has 2 chlorophyll molecules. And so we can start building up completely artificial photosynthetic systems. And we can also now tune the absorbance profile to cover wavelengths which are not well covered by natural photosynthesis. And my final example, again, something that nature never tried to do, is in mineralization. So in nature, we have things like bone and tooth and shells where proteins are directing the mineralization of calcium carbonate, calcium phosphate to make these really remarkable hybrid materials. So we've been really fascinated by this, and so we've been approaching it by designing proteins which have arrays of amino acids that we predict would drive the nucleation and the mineralization of inorganic compounds. In this example, though, we chose not to apply this to calcium carbonate or calcium phosphate, but to zinc oxide, which is a semiconductor. Because you can imagine if you could template semiconductor growth with proteins, that would open up a whole new range of interesting materials. So here we've lined this a part of this structure with this interface, and when we incubate this with zinc oxide, we get zinc oxide filling this part of the triangle, as you can see here, which is opening up really very exciting new ways of making completely new hybrid materials. So I've kind of given you a bit of a whirlwind tour of some of the things that can be done with modern protein design. The applications, there are really a wide range of applications in medicine. I've talked about pandemic preparedness and cancer immunotherapy, neurodegeneration. There's applications in technologies I've showed you, and many interesting problems of sustainability. And my prediction will be that over the next 5 to 10 years, we'll see an increasing number of designed proteins out in the world addressing these problems. Finally, most important part, I want to thank all the amazing people who contributed to this work. It's really, I mean, the prize really celebrates the work of a really large community of people currently in my research group, and then a very, very large community of former students, postdocs, colleagues, and friends. And it's really been just absolutely wonderful to work with everyone. And this prize is really all for everyone who contributed to this. And I'm really happy because I think nearly 200 people have come to Stockholm to help me celebrate this, which is really, really wonderful. And I hope I get some time to actually see them. So thanks.
Thank you for a fantastic lecture. So our next speaker is Demis Hassabis. He was born 1976 in London, United Kingdom, and he received his PhD in 2009 from University College London. And he is presently the CEO of Google DeepMind, also based in London, United Kingdom. So please, Demis, we're looking forward to your Nobel lecture, and please join me in welcoming Dr. Hassabis.
Well, first of all, I want to start by thanking the Royal Swedish Academy of Sciences and the Nobel Committee for the honor of a lifetime. It's so wonderful to see so many family and close friends and colleagues here today. It's been an amazing few days. I'm going to talk about AlphaFold, of course, and the impact it's had so far, but I'm also going to touch on the critical work we did in the lead-up to developing AlphaFold and where I think the future of AI for science is going more broadly. So my journey to AI actually started surprisingly with games, and specifically chess. I started playing chess when I was 4 and played very competitively, captaining many of the England junior chess teams growing up. That's a picture of me on the right playing for board 1 for the England under-11 team when I was 9. And when you play chess seriously at such a young age, it's a very formative experience. And for me, it really got me thinking about thinking itself. I was sort of fascinated, intrigued by how does our mind come up with these ideas, these plans, these strategies, and how can that be improved? And as part of the training camps that we went to, we got actually access to early chess computers, like this one here, the Fidelity Chess Challenger. And some of you remember these early chess computers, you actually had to press the squares on the board physically, the LEDs on the board to move the pieces around. And of course, we were meant to use these machines to improve our chess skills. But I remember actually being fascinated by the fact that someone had actually managed to program this inanimate lump of plastic to play chess well against me. So I was intrigued about this, and I started reading some books about it, and ended up coding my first AI program on my trusty Amiga 500 that you'll see in the middle there when I was 11, but to play the classic game of Othello. And this program, my first AI program, it managed to beat my kid brother. Admittedly, he was only 5 at the time, but I was still amazed that you could make something that could do something sort of independently of you. And that got me hooked on computers and AI for the rest of my life. So fast forwarding then, more than 20 years later, and games are still central to my approach to AI, and actually at the core of our early work at DeepMind. We started DeepMind in 2010 as an industrial research lab with a mission to build artificial general intelligence. And the vision behind it was to create a kind of modern-day Bell Labs. Initially, we used games as the sort of perfect proving ground for AI because it's very easy to generate a lot of data on your computers. You can have the systems play against themselves. And it's easy to specify a clear objective or clear metric to hill climb against. So in most games, you can specify to maximize the score or to just win the game. So it's very clear if you're making progress with your algorithmic ideas. Now, of course, the pinnacle for games AI, which has had a long history, actually from the beginning of AI. So pioneers like Alan Turing and Claude Shannon, they all tried their hands at making and programming chess computers. But the pinnacle for games AI is being Go, the game of Go, the ancient game of Go. It's probably the most complex game we've ever devised. One illustration of how complex Go is that there are 10 to the power 170 possible positions, which is more than there are atoms in the universe. So there's no way you could write a program that could brute force a solution to playing Go well. So our first big breakthrough really was major breakthrough was in 2016 with our AlphaGo system that managed to beat the world champion, the South Korean Grandmaster Lee Sedol 4-1 in a famous match in Seoul. But it didn't just win the match. Importantly, it actually came up with new creative strategies and ideas that had never been seen before, even though we've played Go for thousands of years. And the most famous one of these new strategies was actually Move 37 that I'm showing here in the bottom right. And in game 2 of this challenge match, and it's outlined as the black stone outlined in red. And this move had sort of never been seen before in top-level Go play. And it turned out to be the perfect move to decide this game 2 match 100 moves later, as if AlphaGo had put it there presciently ahead of time. So how did we do this? Well, the AlphaGo system and its successors, AlphaZero, more general successors, mastered the game of Go and also all other two-player games through a process of what we call self-learning. So that's where these systems, instead of being programmed directly with solutions, they learn strategies and solutions for themselves. In this case, by playing many hundreds of thousands, in fact millions of games against itself, and incrementally learning from its mistakes and improving its strategies. And through this process of self-learning, we can build up a useful neural network model of Go. So AlphaGo and this 2016 result was a bit of a watershed moment, I think, in modern AI, as it was the first kind of big proof point that these sorts of systems, these kind of learning systems, were able to do something pretty impressive and thought to be intractable. So with these neural network systems, this neural network model, we can actually efficiently guide the search process so that we don't have to search all of the possibilities in a game like Go or in any kind of complex space. So in this cartoon diagram here, I'm showing a tree of possibilities in a Go game. And if you imagine each node in the tree is a particular Go position, and you have to make a decision about which move to take, and that takes you down this tree. So there's this massive intractable set of possibilities from any particular Go position. And what this neural network model allows you to do, AlphaGo to do, was to only analyze and look at and consider a tiny fraction of all the possible paths. So here, kind of illustrated in the paths in blue. And it does this by basically figuring out what are the most likely moves, what are the most promising moves to look at in any particular position, and also which side, black or white, is likely to win from the current position, and the probability, the estimated probability of winning from that position. And then once the thinking time has run out, say a minute or two, then it outputs the best line it's found so far, the most optimal line it's found so far out of the ones it's considered, out of those blue lines. And here in this case is the line colored in purple. So now that we've sort of cracked the pinnacle of games AI, we sort of knew that we had the techniques and methods to now transport that and tackle important real-world problems, including big challenges in science. So what then makes for a suitable problem for these kind of AI methods to tackle? Well, we look for kind of 3 criteria that make a problem suitable. Number one, finding a path through a massively combinatorial search space. Can the problem be couched in these terms? Secondly, can we specify a clear objective function or metric to optimize or hill climb against? So in games, this is pretty easy. It's winning the game or maximizing the score. And then thirdly, are there a lot of data available to learn the neural network model, and ideally but it could be or, an accurate and efficient simulator to generate more synthetic data in distribution? So it turns out, actually, when you look at problems in this way, there are a lot of real-world problems that actually fit this profile or can be made to fit this profile, including many problems in science. And for me, the number one thing I always wanted to do with these kinds of systems was protein folding. It was always top of my list ever since I first came across it as this fascinating problem as an undergraduate at Cambridge. So as you heard earlier, proteins are the building blocks of life. Almost all biological processes and every living thing depend on proteins for their operation, from the twitching of your muscle fibers to the firing of your neurons. So in essence, you can think of proteins as these kind of exquisite nanoscale biomachines. And they're, of course, extraordinarily beautiful as well. So proteins, as we heard earlier, are specified by their amino acid sequence, which you can see an example of one here on the left. And these sequences fold up into complex 3D structures. For example, this one here folds into this beautiful structure on the right. So you can think of it like a string of beads that kind of scrunch up into a ball. And knowing the 3D structure of a protein tells you a lot about its function, and is of course critical for things like understanding disease and accelerating drug discovery. So the question then is, can the 3D structure of a protein be directly predicted solely from its one-dimensional amino acid sequence? And in his Nobel lecture in 1972, Christian N. Finsen famously conjectured that this should theoretically be possible. And he advanced his thermodynamic hypothesis, the idea that a protein would uniquely take the shape that minimizes the free energy in the system. This then became known as the protein folding problem and kicked off a kind of 50-year quest to find a computational solution to this grand challenge. So why is this problem so hard? Well, of course, the normal way that you would go about finding the structure of a protein is doing it experimentally. And this is very painstaking and difficult work. And it can often take years, months, and even years of work to just determine the structure of even a single protein. And Leventhal actually encapsulated this difficulty very well in what became known as Leventhal's paradox. And he estimated that for a typical size protein, there may be up to 10 to the power 300 possible conformations that that protein can take, a truly astronomical number. So obviously enumerating all of these possibilities would take longer than the age of the universe. And yet somehow, in nature, these proteins fold spontaneously, sometimes in a matter of milliseconds. So that gives us hope, really, that there must be a solution to this, because it must mean that there's some topology in the kind of energy landscape that guides the folding process very efficiently in nature. And perhaps we can recover what that process is. Another key aspect of why we took on this challenge was that there was readily available data to learn from, and also, very importantly for AI development, very clear and great benchmarks to try and measure your progress against. So after decades of experimental work, 170,000 roughly structures had been determined experimentally through lots and lots of painstaking work for tens of thousands of experimentalists, and collated into the Protein Data Bank, the PDB, which is an incredible and valuable resource that we used as the starting point to train AlphaFold. And then secondly, there was the CASP competition, which was considered to be the gold standard benchmark for structure prediction, and was run every 2 years, ever since 1994, by Professor John Moult and his colleagues, and to test the best computational systems. And the beauty of this competition was that it's a blind assessment competition. So the proteins that you're trying to predict computationally the structures have just been discovered experimentally, but have not yet been published. So no one external from those groups knows what those structures are. So it's a true test of whether these computational systems are capable or not. And we're always told that the critical threshold for a computational system like this to be of use to practical use to experimentalists was that you had to get the accuracy down to within the width of an atom. So on average, less than 1 angstrom error, which is incredibly a high degree of accuracy. So with AlphaFold 2, we managed to achieve this atomic accuracy. We started off with AlphaFold 1 back in 2016 and entered it for the first time in CASP 13, the 13th edition of CASP, in 2018. And we really advanced the field here. And if you look at this bar chart of progress, this is basically the winning score for the top team each edition of CASP. As a kind of distance measure of how accurate the prediction is compared to the ground truth. And you can see for about a decade prior, there hadn't been much progress in increasing the accuracy of these predictions. So with AlphaFold 1, it topped the leaderboard of the CASP 13 competition and improved the predictions by a large amount. And it was really the first time that a machine learning system was introduced that had machine learning as the core component of the system. But we still hadn't reached atomic accuracy, which is sort of represented by the 90 GDT score line here on the chart. And we were able to do that by taking the learnings from AlphaFold 1, then re-architecting a brand new system with AlphaFold 2, and entering that into CAS14, and that reached the atomic accuracy needed and led the organizers of the competition to declare the problem essentially solved. So this is the diagram of the innovative architecture of AlphaFold 2. I'm going to leave John to cover some of the technical details of this in his talk, but the high-level thing, that takeaway is that actually there was no silver bullet to solving this problem. AlphaFold 2 actually had to incorporate over a dozen different innovations into quite a complex, what we call a hybrid system. And one of the key things of that was to build in evolutionary and physical constraints into the architecture along with the learning components of the system, and sort of combining those two elements together. And it was key to that the fact of this working is that the AlphaFold team was a multidisciplinary team composed of expert biologists and chemists, as well as machine learning and engineering. And it was a full end-to-end system that started with the amino acid sequence and directly output the 3D structure prediction. And it used this sort of recycling stage that allowed it to iteratively refine the prediction over many iterative steps. So you can see that happening here with this complex protein that was actually in the CAS14 competition. You see it on the left-hand side, the ground truth in green and the prediction in blue, very much overlapping. And then on the right-hand side, you can see how AlphaFold 2 sort of iterates over 192 steps in this case to the final predicted structure, each time improving its accuracy. So then with AlphaFold, once we had the system, of course, we wanted to make the maximum impact possible with it. And AlphaFold is not only very accurate, but it's also very fast. And we sort of quickly realized that it was fast enough to actually practically be able to fold all proteins known to science. So roughly over 200 million proteins that have we know the sequences for, and we would love to know the structures for. And so over the course of the next year, we on our computers, we basically folded all the 200 million proteins. We open sourced AlphaFold, and we built the AlphaFold database with our fantastic colleagues at EBI, the European Bioinformatics Institute, and we provided them free and unrestricted access for everyone to use to all these predicted structures. Of course, we were cognizant about safety and ethics, so we consulted with over 30 biosecurity and bioethics experts before we put the database out to ensure that the benefits far outweighed any risks. And the impact so far has been beyond what we could have imagined. With over 2 million researchers from around the world have made use of AlphaFold and its predictions, over 30,000 citations, and it's very much become a kind of standard tool in the biology toolkit. Just a small sampling of some of the progress that AlphaFold has helped to accelerate across actually a huge range of problems. And some of my favorites, tackling plastic pollution with designing new enzymes, helping with neglected diseases for underfunded diseases that affect a lot of the poorer parts of the world, helping with fundamental structural biology, things like determining the structure of the nuclear pore complex, and even just recently, just a couple of weeks ago, discovering new mechanisms in reproduction. And I think very much we're just at the beginning of the impact that programs like AlphaFold will make. Of course, we've been continuing on with developing AlphaFold, and just earlier this year we released the newest version of AlphaFold, AlphaFold 3. And this is also a big advance because with AlphaFold 2, you can think of it as essentially solving the static picture of a protein and what the structure of it looks like. But of course, we know that biology is incredibly dynamic. And actually, all the interesting things happen in biology when different aspects of biology interact with each other. And AlphaFold 3 is our first step towards modeling the interactions and the dynamics. And it's actually able to model pairwise interactions between proteins and other proteins, but also proteins and RNA, proteins and DNA, and proteins and ligands. So this is a big step towards what one would need to use AlphaFold for things like drug discovery. So I just want to end with a last section on zooming out a bit and talking a little bit about the implications of this kind of work and some of our colleagues on the bigger picture of AI for scientific endeavor. So if we take a step back and look at the essence of what are our systems been doing, both AlphaGo, AlphaFold, and some of the other systems that we've built. And really, we can describe them as finding the optimal solution in this enormous combinatorial search space. And we do that by learning a model of that environment, either from data or from simulation, and then using that model to guide a search process according to some kind of objective function that you're trying to optimize. And it turns out that this is a very general solution and that many problems can fit this approach. So I showed you earlier this tree diagram of finding the best Go move in a Go game. But one could easily switch out those nodes of Go positions for actually chemical compound designs. And now think about the tree of search that you're doing is finding the best molecule or the best drug compound through chemical space. And I think you could very much use the same kind of techniques that I've outlined here for this kind of problem. So that means, if we may be entering perhaps this new era of what I like to call digital biology. I've always thought of biology at its most fundamental level can be thought of as an information processing system, albeit a phenomenally complex and emergent one. And I think it's such a complex system, it's going to be hard to boil down the workings of biology to a few mathematical equations. So I think maths has been an incredible tool to describe sort of description language for physical phenomena, for physics. And I think in the same way, AI may be potentially the perfect description language for biology. And we hope that AlphaFold is a kind of proof point that could, when we look back maybe in 10 or 20 years' time, maybe helped usher in this new era of digital biology. And we're trying to contribute to that, where we recently spun out, a couple of years ago, Isomorphic Labs, a new company to build on AlphaFold and to reimagine the drug discovery process from first principles with AI. And perhaps we can reduce down drug discovery, which is an incredibly difficult and time-consuming and expensive process, down from years to perhaps months, or maybe even one day, weeks. And we sometimes think of this kind of accelerated process that I've just shown you with AlphaFold and the protein structures, and perhaps one day will happen with drug discovery, as sort of doing science at digital speed. And I've had a long-standing dream, actually, to think maybe one day be able to simulate a whole virtual cell, not just a protein or a couple of proteins interacting with each other, but an entire cell, and then those predictions would be useful for experimentalists. So perhaps I'll end then, given this is a Nobel lecture, I thought I would end with a couple of new, perhaps slightly provocative ideas in the spirit of Christian Anfinsen and his 1972 lecture. So ever since doing AlphaGo, actually, I've been thinking a lot about what are the limits of classical systems, classical computing systems. I think there's a big debate going on at the moment in computing circles about quantum computers versus classical systems. And I think classical Turing machines, basically the underpinnings of modern computers today, I think can do a lot more than we probably previously thought. And how can they do that? Well, they do that by perhaps doing this massive amount of pre-compute ahead of time and use that to develop a good model of the environment, a good model of the problem that you're trying to solve. And then you can use this model to efficiently explore solution space in polynomial time, what's called polynomial time in complexity theory, so an efficient amount of time. So I sort of loosely proposed conjecture that I'm thinking about is that maybe any pattern or structure that can be generated or found in nature can be efficiently discovered and modeled by a classical learning algorithm. That doesn't mean everything, all quantum systems, because there'll be lots of things that don't occur in nature that have no pattern or no underlying structure to learn. So for example, factorizing large numbers or abstract problems like that. But I think systems in nature like proteins and perhaps materials will potentially have structure that can be learned by these kinds of processes that I've outlined today. And if it turns out that classical systems then, therefore, can model some types of quantum systems, I think that could have some quite big implications for areas like complexity theory, including P equals MP, and maybe even some aspects of fundamental physics, like information theory. So with that, I just wanted to talk about the final piece, which is that I've talked a lot about AI here for biology and life sciences, but actually we're doing a lot of work at DeepMind across the sciences, medicine, climate, mathematics. And here's just a little selection of some of the work we've done in the last few years, applying some of the AI techniques I've talked about today to medical areas like image analysis and diagnosis of medical images, genetic sequences and spotting missense variations, whether they're benign or pathogenic, controlling the fusion, the plasma in a fusion reactor, coming up with faster algorithms like matrix multiplication, state-of-the-art weather prediction systems, and the discovery of thousands of new materials that we'd never seen before, all using AI as a fundamental tool underlying the sciences. So I think with this, we may be entering a new golden era of discovery, helped by these new types of powerful AI tools. Now, of course, I've worked my whole life on AI because I think it has the incredible potential to help with humanity's greatest challenges. But also, AI is a dual-purpose technology, and it must be built responsibly and safely and be used for the benefit of everyone. And in order to do that, I think it's critical for scientists and technologists at the lead of this field to engage with a wide range of stakeholders, from government to academia and civil society, to incorporate all of the views of how these systems should be deployed and best used. And I think a technology as transformative as AGI will be, I think this will be akin to something like the invention of fire or electricity, requires exceptional care and foresight to steer us through the next stages of the development of this incredibly powerful technology. But if we're able to steward this technology safely through, then I think AGI could end up being the ultimate general purpose tool to help us understand the universe around us and our place in it. So I just want to end, of course, by thanking the amazing and incredible AlphaFold team, almost all of whom are here today, and the AlphaGo and AlphaZero teams, our wider colleagues at DeepMind and Google for all of their incredible support and infrastructure that supports all of this work that you've seen today, our wonderful collaborators at EMBL EBI, the CAS community, and the PDB and experimental biology community. And of course, lastly and most importantly, I want to thank my wonderful family, closest friends and colleagues, all of whom are here today, for their love, support, and encouragement, without which none of this would have been possible. Thank you.
Thank you for a fascinating talk. Our next laureate, our next speaker, is Dr. John Jumper. So John Jumper was born in 1985 in Little Rock, Arkansas, USA. And he received his PhD in 2017 from the University of Chicago, Illinois, also in the United States. And he is at present senior research scientist at Google DeepMind, London, UK. So please join me now in welcoming Dr. John Jumper for his Nobel lecture.
Interesting. This is good. I was a little bit nervous before my lecture, and this should fix it. Nothing. The technical check went excellently yesterday. Metaphor for AlphaFold development. Challenges along the way, but it worked out in the end. So it's absolutely wonderful to be here. It's truly an extraordinary honor to tell you about this work, to tell you about the work of our team in protein structure prediction. And the part that I'm going to focus on today in my lecture is how did we think about building chemistry and physics and biology into this neural network? How did we take AlphaFold from kind of standard machine learning into trying to build the machine learning of proteins, and why did it matter? And kind of a starting point for this, and really the starting point for my entry into biology, is I applied for one job and ended up getting another, and ended up working in this lab that worked on protein simulation, D. Shaw research, and really thinking about how proteins move from a very, very direct kind of brute forcing Newton's equations type of perspective. And there's such a direct way to put the physics into it. And so you think about these atomic systems, this kind of collection of atoms, you kind of throw away the electrons and try and replace them with still a good energy model that can be very, very directly used in order to compute forces, in order to update the positions, and to watch proteins move. And it's absolutely mesmerizing. And it's normally what people kind of think when they say, let us put physics into our protein computation, right? Let us think about the forces on atoms. Let us reduce it to, you know, as a young physicist, right? Your hydrogen atoms, your really well understood kind of physical principles, your van der Waals interactions, your Coulombic interactions. And people write down these kind of equations like you see on the right. This is from Michael Levitt's talk, and really a wonderful depiction of how people think about putting physics into simulation. And we have these kind of nice laws of motion, and if you look at it, you can see things, if you're a physicist, that you really recognize. You say, there is the physics. But if you look really closely, you might realize how much physics is missing. That all of these interactions are pairwise. For example, this is a non-polarizable model. What I love most about the depiction of the equations is that it is almost written in Comic Sans, right? It's written as if it's a kind of coarse and approximate description of the physics, which of course it is, and it's told us a lot. But there is so much more physics. It's really more like a machine learning model of a very particular and effective form, but that is very, very expensive to use. And it is a really interesting way to think about some of the physics, but maybe not others. And it's very, very deeply difficult to reach the types of timescales that we need in order to make predictions of processes like protein folding. And more importantly, even if you can drive one simulation out to the millisecond plus scale on which most folding occurs, you'll find that your model wasn't quite right. Right? This is still a cartoon of the physics in some sense, fit with many parameters. And you need to change those parameters in order to make it more accurate. You need to fit this machine learning model, but it's so very expensive that it's very hard to do so. And so, in fact, if you look at a lot of what has been successful in protein structure prediction prior to AlphaFold and through AlphaFold 1 has been a machine learning point of view, and really in a homology point of view as well, that starts from this amazing resource, starts from the other side from the physics. We start from the observations, and of course it's the protein data bank. And both David and Demis talked about how essential it is, so I'll assume you're convinced by now that this is a tremendous resource, and a tremendous amount of foresight went into it, making it available, collecting the full output of the structural biology community, and so that we can develop computational and test computational methods, and it grows at a rapid pace, and grows at enormous investment. you know, I look at the left-hand side, we've crossed 200,000 structures in the PDB, and each one is something like a year of a student's time, someone that we're going to call doctor at the end of it. It's a truly incredible societal investment in these data, and it gives us another way to go after this. We can, of course, use machine learning, and there have been many approaches to develop machine learning for protein structure prediction. And one of the most successful paradigms would start from the other big source of information we have, protein sequences, would convert that into coevolution statistics, what type of residues varied together, and then use those in order to try and make some predictions about the structure that would then be fed into an engine to kind of turn it into a proper structure. And our AlphaFold 1 system was a very good example along this. And it uses a neural network that was really almost taken off the shelf. I think quite literally the same neural network design as was used for a computer vision project within DeepMind. And what it has is biological inputs, coevolutionary data, sequence data. It has geometric or physical outputs, the distances between proteins. And then, of course, we make many distance predictions, n squared. So for 100 residue protein, we're making, or n squared over 2, we're making 50,000, if I did that math on the fly correctly, 5,000 predictions of protein distances, and then we try and harmonize them through a kind of really, we ended up with a very simple procedure. We had very complex procedures. There's a lot of kind of Leventhal-inspired search, and how do we really do search? Well, it turns out it was kind of gradient descent in the end. And I think what was interesting as we looked into AlphaFold 1, we had built it. It was a great system. It had some new ideas in terms of distances, but there was something actually really interesting and curious about it as we looked deeper. That sometimes AlphaFold 1 was very confident of the distances between different parts of the protein. I think the view going into this, or how coevolutionary data is thought of maybe giving us some information about contacts. It was originally showed that close by residues tend to evolve in a coordinated fashion, and then it was shown that, oh, well, using these data, you can sometimes find out close residues. But AlphaFold 1 would sometimes be incredibly confident that 2 residues were 13 angstroms apart, and that's quite a distance. That's kind of like saying, I am really sure that my friend is 178 meters away, right? You don't know that just by saying, I heard them. You know that because you know exactly where they are, and then you say it's 178 meters. And so similarly, we had some indication, even in AlphaFold 1, that maybe sometimes it was solving local parts of the structure, understanding the geometry really well, and then it was reporting distances by basically measuring the geometry of this thing that it understood in the middle and had no way to tell us. So we had this kind of sign that there was something going on, that maybe these neural networks could do quite a bit more, but we were also held back by, you know, bringing together 2 fields with an awkward interface, right? We had the incredible field of machine learning and all the ideas there, and then kind of in and out, we had, you know, biology, geometry, but we didn't ever meld them. They weren't working together. And because of that, all of the understanding of geometry, biology, et cetera, well, almost all of it had to be learned from data, had to be learned from these, I think at the time, about 140,000 protein structures that we had available. So we were entirely dependent on the general learning capabilities of our general learning systems, and that's great, but that's not very data efficient. How can we take all of this kind of human knowledge that we have and build it into the system? And kind of the first stage of that is we wanted to get to something incredibly simple, not a, you know, you do all this processing and then you do all that processing and you have a little bit of machine learning in the middle. How do we just make it, you know, this simple diagram? We'll start with a sequence on the left, we'll do a big neural network thing in the middle, and then we will end up with a structure on the right. And kind of getting to that really felt like it was going to be essential. We needed some way to do this, and we needed that thing in the middle to be inspired by physics and chemistry and geometry and really build those ideas into the design of the network so that it could be more efficient with the data we had. And the way that we started doing this, and this was really shortly after AlphaFold 1 in late 2018, we started to say, well, let's just throw away the evolutionary information. It's good, but we really want to understand physics. We really want to understand geometry. And so we kind of went back to the beginning. And also, it made our performance on almost all proteins terrible. And starting with terrible performance gives you a license to try weird ideas, because then your weird idea can be state of the art. And the other thing it did is it started to really get us thinking about, well, what is in proteins? What are in these protein chains? And I remember I'd been working with, even back into my PhD thesis, this notion of geometric frames, that each residue has a side chain sticking out of it. And that residue is an important, that's an important thing, where this points. How do we put orientation into it? And we started doing what we would call frames or reference frames. And we started to play originally with side chain prediction. And oh, can you predict where side chains are? And actually, you can do that really, really well with deep learning. And then we kind of thought more and more, well, how much further can we go? And we tried some ideas that looked more like kind of learned simulation. But then we found out, actually, that if you take an architecture that has some understanding of geometry, if you put some of this into the neural network, you can start to produce protein structures directly. You can put in a sequence, and you can say the answer is this structure. And there were a couple of essential innovations along the way. Probably the most important was simply how you measure how correct your answer is. And in fact, we came up with the FAPE loss that says, how are these geometrically and orientationally related? And this started to work reasonably well. And actually, I remember before the first kind of direct structure prediction that worked, I had some idea that that actual folding, making structures, was going to be an imagination for the network. And so if you see the diagram on the bottom, this was essentially the diagram we had at the time, is maybe we'll do embedding, we'll think about the sequence, then we'll do a bunch of folding, and then we'll think about it some more and think about what we got, and we'll do it again and again. It ended up actually looking a lot like recycling, and I didn't appreciate that until we put it into this talk. But we had this idea of we're going to build geometry as a kind of tool that the network is using in producing structures. But what we found is, in fact, that little folding bit already worked really surprisingly well. The answers were still really bad, but they looked like proteins. It was clearly handling the geometry of proteins immediately. And then only a week or two, maybe a month after this started to work, Rich had the idea of, well, we have all of these distance predictions from AlphaFold 1 sitting around. Why don't we just stick those into this geometric structure prediction network as well? And it looked a lot like the structure module, in fact, for the final AlphaFold 2. And so we would stick distograms into this, and almost immediately got predictions that were nearly as good as AlphaFold 1. And so we had nothing but neural network components now. We had kind of stripped at least the geometry off the end and gone to what we knew we needed, the protein structure. But we still had the coevolution, in fact, the fixed network at the beginning. And of course, we tried many, many ideas for new geometric networks, and all of them were about the same accuracy. We pushed it up only a couple of GDT over this research and started to refer to what we called the distogram barrier, that we weren't going to get any better at this problem until we got better at making the distograms, until we eliminated our dependence on the AlphaFold 1 network. And in the same way as we had kind of reimagined how we built and built new components for the network for the end, we wanted to build new components at the beginning. We didn't want it to be dependent on both coevolution and this interpretation of coevolution in the pseudo-likelihood models. To do that, we started over just, and this was actually, we first tried what has now become popular, but we didn't really do big enough or deep enough versions of it, single sequence prediction. Can we just fill in the protein sequence and train models to do that and have that be some sort of way in which the model learns about sequence space and then go and predict distances? But pretty immediately we said, well, the multi-sequence alignment is still great. This still is telling us a lot about the evolutionary history of the protein. We know that it's highly relevant to structure. How can we use this? And so we started to train networks only on this, and in fact do what would be considered mass language modeling training now. We would call it BERT at the time. And we would just put in the raw MSA with a number of blanked out sections and said, guess what amino acid is here? And to do that, you have to understand a lot about both the evolution and the structure in order to guess which amino acids might be in the hidden locations. And this was a raw MSA kind of BERT network, BERT being the first major mass language modeling, so we called it Robert. And we also, I think, made a really consequential choice here, that the multi-sequence alignment, the collection of many, many protein sequences aligned such that each column is an evolutionarily related location, we decided to use axial attention, attention being a way in which the whole sequence can talk to each other in some sort of averaging way. And this was semantic because we could say the sequences meant something. So attending over them is attending over different tips of the evolutionary tree. And the columns meant something different. They meant kind of attending over this position over evolutionary time or across evolutionary endpoints. And alternating those two is a very, very semantic way to think about processing evolution. And this worked extremely well. And so as soon as we had these and they started to work and they looked interesting, we kind of took the same convolutional networks and just basically attached it on top. And so we would run our MSA, it would try and fill in the MSA, and then we would go send on what had been learned to this convolutional network we developed and predict histograms. And this worked exceptionally well. Suddenly this network was learning incredibly quickly. We had broken the histogram barrier. And almost immediately after that, and so of course we would put this into direct folding, but almost immediately after that we said, well, okay, we've gotten some new interpretation of how to process evolutionary data. We've aligned the neural network to do that. Why don't we just attach that to our view of geometry? And so we would put the structure module on it, and we suddenly had end-to-end learning. We would start at almost the raw inputs, the aligned evolutionary data, and then we would produce a structure. Now, we asked it to do two subtasks along the way. Oh, by the way, fill in the MSA. Oh, by the way, make a histogram to keep the network, to give it, lots to think about, lots to do. But we almost immediately had end-to-end structure prediction in kind of early to mid 2019. And it became really clear that this was a great idea, that this also, it meant two things. One is that it was more accurate, it was more aligned. It also meant that it was very, very easy to train and test new ideas. And so we could have this incredibly creative period where we could try many, many ideas and, find out every day that your idea from yesterday was wrong, right? And try each of these. And machine learning is filled with failure. You have intuition and failure, much like lab experimentation. So you need to try many, many ideas to make it work. Now, the other thing we actually realized pretty quickly, and I was reading a paper on sparse transformers on how you can use attention on images, and they said, oh, well, it's a good idea to do axial attention, across the rows, down the columns. And we're sitting and reading that and realized, in fact, that the second part of processing, where we dealt with in-residue by in-residue matrices that represented kind of all pairs in the network that would end up being the histogram, well, in fact, that is a very semantic operation. And if you think of it as a graph, we can attend all of the residues that relate to this one particular, or all of the pairs which attend to this one particular residue in either direction. And this was very semantic. And even better, we had already coded that up because it was good for the MSA. So we started to replace the convolutional networks with axial attention. It was much more aligned also because if you think about pairwise information, well, physics is pairwise only in its energies and only approximately, but the question of structure is highly non-pairwise, right? Things come together. There are many, many pair interactions, and you need all of those talking to each other, communicating. And so axial attention lets them communicate incredibly quickly, lets the network constantly refine its hypothesis rapidly. And the animations that Demis showed of the structure prediction over layers, you can see how quickly it's changing its mind, it's updating its view, and axial attention is essentially what allows that, and that started to really drive enormous accuracy improvements. Now, and I'll only describe a couple of the ideas we used in the middle, but as soon as we had done that, we started to say, well, gee whiz, this process of putting physical and biological intuition into our neural networks is a great idea. Let's do more of it. Let's try all the ideas we can, always guided by what made our structure prediction at the end more accurate. How can we find new ways in order to orchestrate information flows that reflect our understanding of physics, that give the network the opportunity to do something more aligned with our understanding of physics? And so two of the really great ideas one from Michael who said, well, you know, this whole thing of processing the evolution first and then trying to do the structure second, that doesn't entirely make sense, right? You want to think about the evolution in light of the structure, and of course you want to think of the structure in light of the evolution, so why don't we just process them both at the same time? And we have this clever scheme where the evolutionary information, the MSA over here, would come in, and as we were processing the sequences in the MSA, we would bias that information by what we believed about the structure, by making a connection into the structure. Similarly, whenever we learned something new about the MSA, we would drop that back into our representation of pairs or structures, and we created this conversation between what we would call two towers, but I've written it sideways, of the network that allowed it to engage in more precise semantic processing of this. And another idea that was come up with, this was one from Alex, is how do we, you know, well, when we think about processing pairs of residues, right, you have, say, I and J as two residues, and then you might have I and K as another two, and those two are talking to each other. But what about the third edge of the triangle? What if we just put that in? And so we started to put in what we would call triangular attention, and very shortly after triangular multiplicative updates, and let us just build this also because we were inspired by this notion of, well, all of those have a strong relationship. They have the triangle inequality. There's something about all of those distances that must be interrelated. Let's create the connections that let them talk, let them compute about this, and in fact, you can measure afterwards that including these triangular connections makes the final distance predictions more consistent with geometry, about fourfold greater agreement with or reduced violations of the triangle inequality by putting in these connections. And so we're always building, not literally, we're not writing the triangle inequality and forcing the network to use it, because that doesn't work very well. We're building the connections that make it more efficient to learn the things that we know. Now, in fact, in 3 stages of the project, we came to this problem of it's not just about structure prediction, it's also about knowing when you're wrong. And the first time we came up on this was a clever idea of Rich's of, oh, well, let's also train AlphaFold on AlphaFold predicted structures, but just the good ones. And as soon as you say, but just the good ones, you have to say, well, how do I know if a structure is good? And the original answer, and from some other work done by, I think, Tim and David, is, well, why don't we figure out how much AlphaFold's distogram is very particular and confident. And we called this a kind of KL loss. And so we would filter and only use those structures or parts of structures where AlphaFold was really sure of what it was giving. Now, as we got further in the project, in fact, shortly before CASP, we made predictions for SARS-CoV-2. And we said, oh, well, we really need a loss function that tells us, you know, or tells biologists what parts of these predictions can we trust. If we're going to make predictions from SARS-CoV-2 with our, you know, still secret AlphaFold 2 system or almost AlphaFold 2, we better tell them which parts are right. And so, and we said, well, all we really want to know, and thinking about it is, you know, we want to know the correctness, which is typically measured by LDDT, is one of the measures of correctness. And so can we just predict that? And we put almost nothing but a head that says predicts LDDT. And sure enough, that was extremely accurate and became incredibly important after we released. I remember biologists talking about this on Twitter, and they very quickly noticed these confidence measures are really, really reliable, tell you when you should use these structures, and started to say things like, if it's blue, it's true, where blue is our confident color. And it's so important because we can't really afford to have unidentified hallucination. You can have hallucination if you say, I don't think this is right, or maybe this is right. But you really do need to tell biologists who are about to do days, weeks, months of experiments based on what they just saw. You need to tell them if your network believes it. And the final confidence measure actually came up due to a reviewer comment who said, well, predicting LDDT is good and all, but that's a very local measure, and I think TM score is a lot better. Can you predict TM score? And started to sit down and write, no, that's not how this works. And it's like, well, you know, if we predict this and predict these kinds of measures and predict a pairwise measure of whether things are correct, then actually we can tell you what the TM score is predicted to be. And more importantly, this pairwise matrix tells you, you know, a big complex neural network prediction isn't right or wrong. It has a set of hypotheses. Do you really believe these two parts of the protein are near each other? And so we could report that. And I think one of the kind of great and pleasant surprises of the AlphaFold release is how well biologists have used these notions of confidence to inform their research. How much they really do kind of not trust entirely, try and trust in proportion to how it should be used, these systems, and really deploy it responsibly in their research. And I've been so impressed, and they've been so good at that. It was something that I was very worried about. Would they simply trust everything? Right? These are predictions. Now, there are many, many other ideas I haven't told you about. I listed some up here. There are many, many more. Ask the AlphaFold team members in this audience to tell you some great stories. But which parts of it matter? And so after we finished and we were writing our paper, we said, well, can we just knock out ideas and see which ones matter? In the machine learning community, this is called ablations. And so as we moved ideas, I remember we had trouble breaking AlphaFold 2. We found every idea was worth something, and no idea was dominant. And I think this really reflects that there isn't one silver bullet of bringing physical and chemical intuition into the network, that there are many, many ways in which you align it, and every one makes you more efficient. In fact, Mohammed Al-Qureshi's lab trained an AlphaFold 2 reproduction on about 1% of the PDB and found it as accurate as AlphaFold 1. That building these intuitions into the network is about worth 100-fold in data. So we are amplifying our usage of the precious experimental data we have by 100-fold by building these ideas in, by doing new types of machine learning. We, of course, make this available in partnership with EBI, who've been incredible partners in making a huge number of structure predictions available, enabling new research. I love research, for example, that finds an unknown density in a experimental cryo-EM structure and then searches through AlphaFold database to find, oh, well, this protein matches in shape and confirms it with biochemical experiments. There's so much more we can do, and we're ultimately closing what has been an enormous gap, that protein sequences are discovered at about 3,000 times faster than protein structures are determined. And we need machine learning to close that gap, and also to bring kind of the importance of protein structure not just to humans, but people that study plants, people that study pathogens, all of those different underinvested areas. And I think it's so exciting to see people use it. Now, finally, and I want to acknowledge very, very much, this is the work of a team and the best team that I've ever worked with. And I decided not to use the pandemic one of us all as squares on Zoom, but for most of the work, it was shoulder to shoulder. And these are really a wonderful group of people, and if you get a chance, ask them to tell you some of the stories. They are truly wonderful. And I want to acknowledge everyone who contributed. There was really a huge effort across many areas. The core team was about 15 or 18, but the support we got from the rest of DeepMind, from the wider community, was exceptional. Our collaborators, of course, at EBI, CASP, who not just kind of assessed and showed the world how you should assess computational science, but really helped to define the problem. You know, brought us GDT and other measures that really, really matter, and our research is deeply indebted to them, showing what works and showing how you should measure it. And of course, the experimental community, and I hope some of the people in this room on whose structures we trained. Thank you.
Okay, please join me now in welcoming all three chemistry laureates up on stage.